For more than a century, the story of early modern humans has seemed relatively clear. Our species emerged in Africa, gradually developed the traits we recognize as fully human, and then spread outward, replacing earlier human populations as we moved into new lands. Fossils, stone tools, and archaeological sites appeared to confirm this picture. Africa was seen as the singular cradle. Migration was imagined as a straightforward expansion, and the origins of modern humans felt, if not simple, at least stable. This understanding shaped textbooks, documentastes, and suggested a linear path from early ancestors to us, with clear boundaries between species and populations. Neanderthals belonged to Europe and Western Denisovans to distant Asia, while early modern humans were thought to remain largely separate until the very end of their journeys, replacement, and survival through superiority, or innovation. But this clarity was in many ways an illusion, created by limited evidence. Entire populations can vanish from the archaeological record without leaving a trace for decades. Researchers worked with fragments of skulls, scattered tools, and indirect clues, building the best narrative they could from what survived. What could not be seen was quietly ignored. Over the last 2 decades, a new kind of evidence has emerged, one that does not rely on the shape of a skull or the style of a tool. Locked inside ancient bones and teeth, fragments of genetic material have survived across tens of thousands of years. Ancient DNA has made it possible to hear biological voices. This genetic evidence does not simply add detail to the old story. It changes its structure. It reveals unexpected connections between populations once considered separate. It exposes migrations that left little archaeological trace. And it shows that early modern humans were shaped not by a single origin or a clean expansion, but by repeated movements, encounters, and moments of near extinction. As ancient DNA began to be recovered and decoded, the origins of early modern humans became more complex, more human, and far less predictable than anyone had imagined. The story we thought we knew is no longer sufficient. And to understand why, we need to return to the assumptions that once defined our past, before the genetic evidence forced us to see it differently. Before ancient DNA reshaped the field, the story of early modern humans rested almost entirely on physical remains. Archaeology and paleoanthropology were the primary tools for reconstructing deep history. From stone tools scattered across landscapes to fragments of skulls pulled from caves and riverbeds, researchers assembled a narrative piece by piece, guided by form, location, and age. The dominant model that emerged during the late 20th century was often described as out of Africa. According to this view, anatomically modern humans evolved in Africa. Roughly 200 ACE developed traits such as lighter skeletal frames, rounded skulls, and complex symbolic behavior. After tens of thousands of years of regional development, within Africa, a small group began to migrate outward, Eventura, Momon-Bire. In this framework, earlier human groups, such as Neanderthals and Denisovans, were seen as evolutionary side branches. Neanderthals occupied Europe and parts of Western Asia, while Denisovans were inferred from limited remains in Eastern Asia. These populations were considered biologically distinct and largely isolated. When early modern humans arrived in their territories, the prevailing assumption was replacement rather than interaction. The older group has disappeared, and modern humans took their place. This interpretation was reinforced by the physical differences in fossils. Neanderthals had robust bones, prominent brow ridges. Early modern humans appeared more gracile and flexible in form. The contrast encouraged a clean division between us and them, suggesting that coexistence, if it occurred at all, was brief and biologically insignificant. Migration, it bifurcated once or twice, followed coastlines or river valleys, and gradually felled empty landspace. Maps showed arrows moving steadily outward, with little indication of backtracking, fragmentation, or failure. Population sizes were often assumed to be large enough to sustain continuous expansion, and extinction was framed as something that happened to others, not to our own ancestors. Within Africa, the picture was even more limited. As a result, Africa was often treated as a single evolutionary stage rather than a complex mosaic of populations. Regional differences were acknowledged, but they were rarely seen as deeply significant. The idea that early modern humans could have been divided into long separated groups within Africa itself, without genetic data. There was little way to test these assumptions. Similar-looking skulls were grouped together. Tool styles were linked to cultural continuity. Gaps in the record were filled with cautious speculation. When contradictions appeared, they were often explained away as low-variation. Researchers worked within the limits of what could be observed directly, but those limits shaped the story in powerful ways. They encouraged simplicity, linearity, and separation. They favored models where change happened gradually and predictably, and where the origins of modern humans could be traced back to a single identifiable source. By the early 2000s, this narrative had become deeply embedded. It was taught as a settled framework rather than a working hypothesis. Yet even then, signs of tension were visible. Some fossils did not fit neatly into categories. Certain tools appeared earlier or farther from expected origin, and questions lingered about how small scattered populations could have survived the environmental instability of the Ice Age world. These unresolved issues were not yet enough to overturn the established view, but they created space for doubt, and when genetic material began to emerge from ancient remains, it would reveal that many of the assumptions underlying the old story were incomplete, and in some cases, fundamentally wrong. The transformation began quietly, in laboratories, rather than excavation sites. For decades, the idea of recovering DNA from ancient human remains was considered degrades rapidly after death. By the time bones fossilize, DNA is usually long gone. Early attempts in the 1980s and 1990s were plagued by contamination and false results, reinforcing skepticism across the field. What changed was not a single discovery but a series of technical breakthroughs. Researchers learned that dense bones, especially the petrous portion of the temporal bone near the inner ear, could preserve DNA far better than other skeletal elements. Teeth, protected by enamel, also proved to be valuable genetic reservoirs. At the same time, clean room techniques reduced contamination, while new sewing technologies made it possible to read even tiny damaged fragments of genetic code. Scientists began working with millions of short DNA fragments, comparing patterns across populations. Statistical methods allowed them to distinguish ancient genetic signals from modern contamination. What emerged was not a complete picture all at once, but a growing genetic map of ancient humanity. One of the first major breakthroughs came from Neanderthal remains. When researchers successfully sequenced substantial portions of the Neanderthal genome, instead of being completely separate from modern humans, Neanderthals shared clear genetic. This overlap was not random. It followed geographic patterns. Strongest in populations outside Africa, this finding alone challenged a central assumption of earlier models. It showed that early modern humans and Neanderthals had not simply replaced one another. They had met, interacted, and exception. It was part of the human story. Soon after, genetic material from a small fragment of bone found in a Siberian cave revealed something even more surprising. The DNA did not match Neanderthals or modern humans. It represented a previously unknown. Nothing was known about their physical appearance, but their genetic legacy was unmistakable. Traces of Denisovans were found in modern populations in South Asia, Australia, and the Pacific. This discovery reshaped how scientists thought about ancient populations. It became clear that the human family tree was not a simple branching diagram, but a net-braided, reconnected, and exchanged genes across vast distances and long periods of time. As techniques improved, researchers pushed further back in time and deeper into Africa. Ancient DNA from early modern human remains, dating to roughly 40 to 50,000 years ago, revealed unexpected diversity. Some African populations showed deep genetic splits, Others carried genetic signals suggesting contact with now extinct human groups that left no fossil record. Equally important were findings related to population size. Genetic patterns revealed repeated bottlenecks. Periods when human populations shrank dramatically. These bottlenecks were not always visible archaeologically. They suggested that early modern humans were often vulnerable, fragmented, and at risk of extinction. Dates and locations began to shift as well. Genetic evidence indicated that humans may have left Africa earlier than previously thought, possibly in multiple waves rather than a single expansion. Some of these early migrations may have failed, leaving little trace except faint genetic echoes in later populations. What made ancient DNA so powerful was not that it replaced archaeology, but that it exposed what archaeology alone could not see. It revealed relationships between populations separated by thousands of kilometers, and it provided a direct record of ancestry, survival, and loss. By the time dozens of ancient germs had been sequenced, the implications were unavoidable. The traditional narrative of human origins could no longer stand on its own. The genetic evidence was pointing toward a far more complex reality, one shaped by repeated migrations, deep population structure, and encounters between different kinds of humans. And as the data accumulated, it began to reveal exactly how different that reality was. As ancient DNA evidence accumulated, patterns began to emerge that fundamentally changed. One of the most important revelations was that modern humans did not arise from a single uniform population. Instead, they emerged from a web of deeply structured groups, some of which had been separated for tens of thousands of years before reconnecting. Within Africa, genetic data revealed that early modern humans were divided into multitudinary paths. These groups were not simply regional variants of the same people. They were genetically distinct lineages, shaped by geography, climate, and isolation. Some lived in wetter environments, others in arid or forested regions. Over time, they accumulated unique genetic traits, while still belonging to the same broader species, rather than one birthplace. Africa contains several long-standing population centers. Net divergence between some of these groups rivals or exceeds the differences seen between modern continental populations. There was no single original genome carried outward. Migration exported variation, not uniformity. Ancient DNA also showed that early expansions out of Africa were neither clean nor final. Genetic signals indicate multiple dispersals, some occurring earlier than previously believed. Some of these early movements may have reached parts of the Middle East and Asia but later disappeared, leaving no direct descendants. Others contributed small but measurable genetic traces to later populations. Interbreeding emerged as a recurring theme. Neanderthal DNA present in all non-African populations. This interbreeding likely occurred soon after modern humans left Africa, and again as populations spread across Eurasia. The genetic contribution was not evenly distributed, some reasoned, show higher levels, suggesting Baroxian. Denisovan ancestry revealed an even more complex picture, populations in islands Southeast Asia, New Guinea. This indicates encounters between early modern humans and Denisovans. Far from the Siberian cave where Denisovan died, it suggests Denisovans once occupied a wide range across Asia, adapting to diverse environments, before disappearing as a distinct population. Genetic data also uncovered evidence of contact with unknown human groups. In Africa especially, some populations carry DNA segments that do not match any known ancient genome. These segments appear to come from deeply divergent hominin lineages that split from the human line hundreds of thousands of years earlier. These groups left little or no fossil evidence, yet their genetic legacy for range. DNA revealed moments when human numbers dropped sharply, sometimes to just a few thousand breeding individuals. These events likely coincided with climatic instability, droughts, or volcanic events. Bottlenecks reduced genetic diversity and reshaped populations, amplifying certain traits while erasing others, or during major migrations out of Africa. This means that many non-African populations descend from a relatively small subset of African diversity. The genetic similarities shared by people across Europe, Asia, and the Americas reflect this narrow ancestral base rather than recent connection. Extinction, once thought to be something that happened mainly to other human species, was revealed as a constant threat to early modern humans themselves. Some lineages vanished entirely, others survived only through absorption. Ancient DNA makes clear that survival was not guaranteed, and that the humans alive today represent only a fraction of those who once existed. Taken together, these findings dismantle the idea of a simple replacement or a single successful expansion. Early modern human history was shaped by repeated movement, fragmentation, reunion, and loss. The boundaries between populations, or porous, and fluidocroissant, was neither straight nor inevitable. What ancient DNA ultimately revealed is that modern humans are not the product of isolation, but of connection. Our origins lie not in one place or moment, but in a long process of interaction among diverse human groups, navigating a changing world. The discoveries revealed by ancient DNA, they change how we understand ourselves as a species. Early modern humans were not a singular group moving confidently toward global dominance. They were vulnerable populations, divided by geography, climate, and chance, surviving through connection rather than isolation. The genetic material carried by people today is not solely inherited from one ancestral population. It is a mosaic shaped by encounters with multiple human groups, some of whom no longer exist as distinct populations. Neanderthals, Denisovans, and unknown African hominins are not distant curiosities. They are part of the biological history carried within living humans. This understanding dissolves the sharp boundaries that once separated modern from archaic. The distinction remains useful for classification, but it no longer implies separation in lived reality. Early modern humans were part of a broader human landscape, one in which interaction, learning, and interbreeding occurred repeatedly. Cultural exchange likely accompanied genetic exchange. Even if much of that cultural evidence has been lost, ancient DNA also reframes the idea of human success. Survival was not driven by superiority in a simple sense. Many populations with advanced tools, or cooperation, and resilience during periods of extreme environmental stress. Bottlenecks show that survival often came down to chance, as much as innovation. The genetic evidence highlights how small and fragile early human populations really were. At several points in our history, the entire species may have been reduced to numbers that would today be considered endangered. That modern humans survived at all is not evidence of inevitability, but of persistence through repeated crises. Another consequence lies in how we view human diversity today. Differences among modern populations are shallow compared to the deep genetic divisions that once existed. What remains is a shared inheritance, shaped by ancient mixing and recent expansion. This perspective does not diminish the uniqueness of human cultures or histories. Instead, it places them within a longer continuum. The boundaries that matter most in the deep past are not those between timelines or modern identities, but those between survival. Ancient DNA does not tell us who we should be, but it shows clearly who we have always been. A species shaped by movement, interaction, and shared ancestry. The humans of the present are not the descendants of a single successful group, but the result of countless lives that intersected, merged, and endured across time. The story revealed by ancient DNA is not dramatic in the sense of sudden revolutions or singular origins. It is quieter, more complex, and ultimately more grounded in evidence. It shows that early modern humans did not appear fully formed and move confidently into the world. They emerged gradually within Africa. From a network of populations shaped by isolation and reunion, their history was marked by movement, contact, and repeated vulnerability. Ancient DNA has allowed researchers to move beyond surface similarities and visible remains. It has provided a record of ancestry written at the molecular level, one that preserves relationships even when bones and tools are gone. Through this record, we see that early modern humans interacted with other human groups wherever they went, adapting to new environments and surviving events that could easily have erased them. What makes this evidence especially important is its restraint. Ancient DNA does not offer a single origin story or a final answer. Instead, it constrains what is possible. It shows where populations split and rejoicably connected different groups truly were. It replaces assumption with measurable relationship and speculation. In doing so, it reshapes our understanding of what it means to be human. Modern humanity is not the endpoint of a straight evolutionary line. It is the result of persistence across instability, of shared ancestry, a connection rather than separation. The origins of early modern humans are not found in one place or moment, but in a long history of movement, encounter, and continuity. Ancient DNA continues to refine this picture as new samples are recovered and new methods are developed. Each genome adds clarity, but also humility. The deeper we look, the more evident it becomes that our past is larger than any single narrative. What remains constant is the value of evidence and the quiet power of genetic traces to illuminate lives lived tens of thousands of years ago.